I would like to present Tanja Jesenko. She comes from the Institute of Oncology in Ljubljana. She will talk about the upregulation of DNA sensing pathway can mediate the off-target effect of radiotherapy. Please, Tanja, take your time.
Yeah.
Okay, thanks.
Thank you for the introduction. Yes, today I will present our research about the DNA sensing pathways and their involvement in the off-target effects of radiotherapy. Radiotherapy is a mainstay of cancer treatment, with approximately one-half of all cancer patients receiving it as a part of their standard of care treatment. Its target anti-tumor effects include the direct action of photons inducing damage in the DNA, and also indirect damage, which is induced by the generation of free radicals that cause the damage on the DNA. And for several decades, the central dogma in the field of radiotherapy was that radiation effects are restricted only to the irradiated cells. First out-of-field or off-target effects in humans were reported back in the 50s. However, the first study demonstrating that radiation damage can be detected also in non-irradiated cells neighboring the irradiated cells was published almost 40 years later. The effect is now called radiation-induced bystander effect and refers to the diverse biological phenomena occurring in non-irradiated cells neighboring the irradiated cells because of the signal transmission from the irradiated cells. It can be mediated by different soluble factors such as cytokines, reactive oxygen species, and others, and can be transported also extracellularly by soluble factors or vesicles, or also by gap junctions. The bystander effect is important because it's implicated in the potentiation of the local tumor response, and also it's involved in the normal tissue damage. The second off-target effect of radiotherapy is an abscopal effect, and this refers to the action at a distance from the irradiated volume, but within the same organism. In the era of immunotherapy with immune checkpoint inhibitors, the abscopal effect is gaining more attention, as it was demonstrated that radiotherapy can mediate systemic rejection of cancer in conjunction with immune checkpoint inhibitors. The mechanisms of the radiation-induced abscopal effect remain unclear. However, the immune system is recognized as its primary mediator. One of the molecular pathways that was implicated in the abscopal effect of radiotherapy is the cGAS-STING pathway. This is a DNA sensing pathway. cGAS is a cytosolic enzyme that, after the binding of DNA, catalyzes the production of the cyclic GAMP that binds STING, which then translocates to the perinuclear structures and induce the production of immunostimulating cytokines, especially type 1 interferon. This pathway is a part of the evolutionary conserved pathways mediating immune response against viruses, which inject also the nucleic acids in the cytosol of cells. And similar to viral infection, radiation was demonstrated to induce the accumulation of DNA in the cytosol of tumor cells, which can be detected by the cGAS-STING pathway, and this results in the production of type 1 interferons that can mediate the abscopal effect by the systemic rejection of distant tumors by generation of the anti-tumor CD8 lymphocytes. However, besides the cGAS-STING pathway, multiple DNA sensing pathways exist as an antiviral defense mechanisms. Cytosolic DNA sensors can be of different types of DNA binding proteins, such as helicases, pyrene and hinge domain family, nucleases, and also enzymes and others. But all of them can induce the activation of specific molecular pathways that induce the production of various cytokines that breach the innate and adaptive anti-tumor immunity. Therefore, our experimental question was, are there also other DNA sensing pathways involved in radiation response that can contribute to the abscopal effect? And also, are the DNA sensing pathways involved also in radiation-induced bystander effect? Our experimental approach was an in vitro approach in order to compare the effects of radiation in two model cell lines which are abundant in the microenvironment of solid tumors. For the tumor cells, we use the B16F10 mouse melanoma cells, and for the macrophages, we use the Ro264.7 mouse macrophages. We irradiated both cell types with different doses of ionizing radiation and followed the survival of these cells in the early time points after irradiation with Presto Blue viability assay. We conducted gene expression studies of the DNA sensors and cytokines by quantitative real-time PCR and used imaging studies for the following the activation of the different sensors and transcription factors and by staining with immunofluorescence. And for the cytokine protein expression, we use the flow cytometry. We first determined the survival of tumor cells and macrophages in the first 3 days after irradiation in order to determine the events that take place before the irradiation-induced cell death. Cell survival was significantly reduced only at 72 hours after irradiation in tumor cells. Macrophages were more radiosensitive than tumor cells. Their survival was reduced already 48 hours after irradiation. Therefore, 24 and 48-hour time points were selected for the rest of the experiments in order to evaluate the effects that take place in irradiated but still viable tumor cells. In these early time points, we evaluated if DNA accumulates in the cytosol of both cell types. We observed a significant increase in the number of micronuclei after irradiation of tumor cells at 24 and 48 hours after irradiation. A similar but even more potent response was observed in macrophages, where the even more increased number of the micronuclei per cell was detected. And I want to point out that in the imaging studies, macrophages were followed for only 24 hours after irradiation due to their increased radiosensitivity and evident cell death at 48 hours. Then we wanted to evaluate also other types of the sources of DNA, which would be the smaller DNA fragments as seen on under the microscope as the DNA spots. We stained the cells with the antibodies against double-stranded DNA, and the results showed that the number of double-stranded DNA spots per cell significantly increased 24 and 48 hours irradiation in tumor cells. And similar as in micronuclei detection, a more potent response was observed in macrophages. So there was increased number of DNA spots in macrophages compared to tumor cells. Okay, so now we established that the radiation induces the accumulation of two types of sources of DNA, which are the micronuclei and also DNA fragments seen as the DNA spots. And now the next question is, which of the DNA sensors do bind and recognize this DNA in the cytosol of cells? For this reason, we conducted the expression studies and evaluated the upregulation expression of different DNA sensors. And the results showed that only 3 of the sensors were upregulated, which were DDX60, DI, and P204 in tumor cells. In general, the expression increased with the time and also with the delivered dose. So in higher doses delivered, there were significantly greater induction of the expression of the DNA sensors in tumor cells. Similar response was observed in macrophages with the same 3 sensors, DDX60, DI, and P204 being upregulated after the irradiation, and also one additional DNA sensor, RIC1, was also upregulated. Similar as in DNA, this upregulation was more potent in macrophages as in tumor cells. So these expression studies showed that higher doses of radiation are more potent to induce the DNA sensors upregulation, as seen for the 6 and 8 grays, but not at 2 gray irradiation, which is a standard dose for the conventional radiotherapy. What was interesting was that STING was not upregulated in our study. Therefore, we used the immunofluorescent staining in order to evaluate its activation. And after the staining of STING, we detected that the STING translocates from the cytosol to the perinuclear punctate structures, and the fraction of cells with the STING perinuclear staining significantly increased after 24 and 48 hours after irradiation of tumor cells. And the similar response was observed also in macrophages. So the STING was not upregulated, but it still is activated upon radiation. Now, now we established that the DNA that is in the cytosol of cells is binded by the DDX60, P204, DI, and STING in tumor cells, and that one additional DNA sensors is upregulated in the macrophages. And next, we were interested in finding out which of the transcription factors are activated and translocated to the nucleus due to the activation of these DNA sensing pathways. We determined the translocation and activation of 3 different transcription factors. The first was interferon response factor 3, so IRF3, that was significantly translocated from the cytoplasm to the nucleus, so it was activated in tumor cells upon radiation. And the similar response was observed in macrophages, so that in both cell types, the IRF3 transcription factor is activated upon radiation. The second was IRF7, which was, on the contrary, not translocated to the nucleus significantly after radiation of the tumor cells, but the potent translocation was observed in macrophages. So the IRF7 transcription factor is only activated in macrophages. The last was the NF-kappa beta, which was evaluated, and the results showed that in tumor cells and also in macrophages, the NF-kappa beta transcription factor is activated upon irradiation. So now we we determined the signaling pathways of the DNA sensing pathways in the tumor cells and macrophages with the transcription factors NF-kappa beta and IRF3 being activated in the tumor cells, and also IRF7 additionally in the macrophages. And the next question is, which cytokines are then produced in response to the activation of the specific transcription factor in both cell types? We follow the expression and the production of cytokines and determined that in tumor cells, interferon beta and tumor necrosis factor alpha are produced in response to these pathways. And in macrophages, there was also interferon beta activated, but also interleukin-1 beta. So now we evaluated and identified the distinct signaling pathways are involved in response to DNA sensing that is cell type dependent, and also the production of the cytokines is cell type dependent, TNF-alpha and interferon beta in tumor cells, and interleukin-1 beta and interferon beta in macrophages. And because all of these cytokines can have the direct cytotoxic effect, the next question is, can these cytokines mediate also the radiation-induced bystander cell effect in the bystander cells? For this reason, we conducted some of the medium transfer experiments. We irradiated the tumor cells or macrophages with 6-gray irradiation, and then 24 hours after irradiation, we harvested the medium and transferred it to the fresh non-irradiated tumor cells, and that followed the survival of tumor cells with the clonogenic assay. The results show that if we transfer the irradiated medium from the tumor cells, irradiated tumor cells to fresh tumor cells, a cytotoxic effect can be observed, and similar was observed when the medium from the irradiated macrophages was transferred to the fresh tumor cells. In conclusions, the conclusions are that radiation induced the accumulation of the DNA in the cytosol of tumor cells and also macrophages in the form of micronuclei and also DNA spots or smaller fragments. That cytosolic DNA can be sensed by distinct DNA sensor, which is dependent on the cell type. That the upregulation of DNA sensors is more potent when higher doses are used, such as in stereotactic body radiotherapy, and not in the 2 gray fractions which are used in the conventional radiotherapy. That the effect is more potent in macrophages as in tumor cells, and that the resulting cell-dependent production of cytokines can mediate the radiation-induced bystander effect. So for the take-home message, we can say that high-dose irradiation activates the DNA sensing pathways, and that this is an important mechanism mediating the induction of stimulatory cytokines, which can mediate bystander and abscopal effect in solid tumors. And that the activation of the DNA sensing pathways differs based on the cell type and is also time and radiation dose dependent. Therefore, every cell type in the microenvironment of solid tumors may have its unique response to irradiation, depending on the combination of the activated DNA sensors and the subsequent pathways. Therefore, the understanding of the timing of the activation of these pathways and subsequent cytokine expression, which are induced by different radiation doses, could improve the schedules for combining immune checkpoint inhibitors or other immunotherapies with the radiation therapy. At the end, I want to thank all of my colleagues at the Department of Medical Oncology, Institute of Oncology, Ljubljana, and also Professor Heller from the University of South Florida, and also the Slovenian Research Agency and National Cancer Institute for funding this research. So thanks for your attention.
Thank you, Tanja. Great talk. This shows how more complex is all the picture. And one question is if you already check also on the resistance cell to radiotherapy, how this pathway is also changed, the distinct pathway if it changes in the radioresistant cell.
No, unfortunately not yet, but this is a great idea. Yeah.
Okay. Okay, from Tiomir Todorov, thank you for the presentation. What are the radiation agents that you used? Do you think you might observe different factors when alpha, beta, or gamma emitters are used?
Oh yes, there are some studies that different radiation sources induce different responses biological in the cell. So yes, this I think that the effect would be different probably. We use the X-ray radiation.
Okay, thank you. So there are not other questions at the moment, so we can go ahead. Thank you.